Hi everyone, so today I'm just going to run you through our Cure Parkinson's funded project looking at repurposing anti-gout medication for Parkinson's. I'm Dr. Rachel Hughes, and this work has been in collaboration with a few different people at the University of Sheffield and at Motac Neuroscience Limited. So I'm not going to go through all the ins and outs of Parkinson's, as I'm sure a lot of you watching will have quite a good understanding. But what I will say is that the prevalence of Parkinson's is increasing, and the current treatment options only treat the symptoms. They don't alter the course of the condition. So that means there is a great need to identify new drug compounds to treat Parkinson's. So the big question is, how do we do this? So there's a few different ways to identify new compounds to treat any condition, including Parkinson's. New and existing compounds can be screened against known targets and deficits in cells from people with Parkinson's. So we can screen new novel compounds, which are usually generated by industrial companies. However, we have no existing safety data for these. Alternatively, we can look at compounds that are already on the market for the treatment of different conditions, and then see if they would be useful in treating things like Parkinson's. So these are known as repurposed compounds, and the major advantage of these is that we already have human safety data. So the mitochondria in Parkinson's, in our lab, we're interested in a particular part of the cell called the mitochondria. And you can think about these kind of like the batteries of a cell, and they provide the energy that a cell needs to survive. So when we're young, our mitochondria function really well. They're able to provide a lot of energy to the cell. However, as we get older, the mitochondria become less efficient and they aren't able to provide as much energy. However, there is still enough for the cells to survive and function correctly. But in conditions such as Parkinson's, the mitochondria become even less efficient and are not able to produce enough energy to successfully support the cell. And so brain cells particularly require a lot of energy to survive. And if too little energy is provided by the mitochondria, then the brain cells can't work properly and they will eventually die. And we see this in the brain of people with Parkinson's. So the work leading up to this project. So in our lab, we generate brain cells and study the mitochondria, and we can look at different measures of mitochondrial health. And to do this, we take small skin biopsies from people with Parkinson's from their forearm. We can then grow them in flasks as a type of skin cell called a fibroblast. These can then be reprogrammed into a type of stem cell called a neuronal progenitor cell. And then through a 3-stage protocol over 27 days, we can differentiate them into the type of brain cell that is affected in Parkinson's. So these are called dopaminergic neurons. And a previous screen in our lab looking at repurposed compounds found 2 compounds in particular that have beneficial effects in cells from people with Parkinson's. And both of these compounds are currently used to treat gout. So from the initial screen, in these graphs, we have 3 different causes of Parkinson's. So in red is cells from people with Parkinson's of an unknown course, otherwise known as sporadic, and then 2 different familial forms of Parkinson's. And when we look at 2 specific markers of mitochondrial health, mitochondrial membrane potential and ATP, we can see that these are decreased compared to people without Parkinson's, which are the bars in blue. However, when we treat with our compounds, we see an improvement in these parameters. And we also see a similar thing when we look at the recycling system. So when mitochondria are damaged, they're recycled in a process known as mitophagy. And again, in the 3 causes of Parkinson's that we're looking at here, they all have problems in their mitochondrial recycling system or mitophagy. And again, our compounds are improving this. So what we now want to know is, how are these compounds working? So we have an idea and we've identified what we think is the target of these compounds. So now we need to prove it. And this is the main crux of this project. So we've identified 4 aims. So first, to confirm the target via a knockout. So we're removing the target from our cells. We also want to investigate the different mechanisms that the target is involved with in the cells. We want to look at neuronal morphology, so what the cells look like, and also the survival of the cells following treatment with the compounds. And we also want to look at the compounds in a mouse study. So this is the work that is being carried out at Motac Neuroscience in France, and they're going to ship the tissue to us for assessment. So I'm going to go through each aim individually. First one, looking at how we're going to confirm the target via knockout. So you'll recognise this first schematic from earlier on in the presentation. But this time we want to remove the target from our cells. So we get to the stem cell or the IMPC stage. And here we add a special virus that can cut the DNA at a certain place. This has then removed the information about our target from the DNA of the cells, and we can then differentiate them into the type of brain cell affected in Parkinson's and see what effects this has. So what we would expect to see is that there would be a loss of the compound effect when we look at the recycling system, the mitophagy. So far, this project only started in May. So far I have treated the stem cells with the virus, and I'm growing the cells up and banking them so that then after Christmas I can start the differentiations and have a look at mitophagy. In the meantime, I've also been looking at a knockdown of our target in our end stage brain cells. So this means that the target is still present within our cells, but at a much reduced level. And again, we do this by adding a virus to the end stage brain cells, and this virus contains something called shRNA. So like I said, I've begun treating these end stage brain cells with the shRNA and looked at the expression of our target. And this is the bars in these graphs in pink. And as you can see, compared to the blue bar, we're seeing much less expression of our target, which is what we wanted and what we expected to see. So I'll now go on to see how this affects the recycling system when I treat with our compounds. So the second aim is to investigate different mechanisms that the target is involved in. So to do this, this is an example of a Western blot on the screen, and this looks at protein expression. So using this, I can look at whether the protein expression of our target and other proteins that it interacts with are different between people with Parkinson's and people without Parkinson's. I can then treat with our compounds and see if anything changes here. To look at the morphology or how the cells look and the survival, it's important for us to have this information and we can see how they survive. So this is an example of the type of images that I get when I image the cells on our high content imager. Work is currently ongoing. I don't have any update on this, but the experiments are in progress and I'll be able to update in due course. This is just an example of some images. And then finally, to assess the compound effect in a mouse model. So this work, like I said, is being carried out at Motac Neuroscience Limited. And we want to see if our compounds are firstly safe in an animal model of Parkinson's and whether they improve any of the features of Parkinson's. So in Parkinson's, we see clumping of a protein called alpha-synuclein in the brain. And these clumps go on to form Lewy bodies. And at Motac, they take these Lewy bodies from the brains of people with Parkinson's that have donated their brains when they pass away and inject them into the brain of a healthy mouse. And this mimics how the alpha-synuclein spreads through the brain of people with Parkinson's. So once they've done this, they then treat the mouse with our compounds and will assess whether the compounds have any beneficial effects. So they will ship the tissue to us in due course and we can have a look. So to conclude, work is ongoing to confirm the target of our repurposed drug compounds. The tissue from the mouse study will be shipped soon and we can have a look at the effect of the compounds on a full biological organism. And in every stage here, we're using patient-derived samples in all aspects of the study. So we hope that this means that it's then better translated into the clinic and that these compounds can then enter preclinical testing when this study concludes. Thank you.